Hello, my name is Maeve King and I'm currently a PhD student in Rich Ivory's lab at UC Berkeley. I'm excited today to contribute to this panel by discussing the challenges that come with imaging the human cerebellum, along with the advances that have been made to better understand the functional role of this underappreciated structure. So I spend a lot of my time thinking about the other brain that's missing from this picture. And by other brain, I of course mean the little brain or the cerebellum. And this is the brain structure that is at worst excluded from the field of view of many fMRI studies and at best relegated to the supplementary materials. However, as many of you know, although only comprising 10% of the total volume of the brain, it's actually home to approximately 80% of the total number of neurons in the human brain. And this simply makes it too big to ignore. So given how different cerebellar architecture is from cortical architecture, I first want to spend a bit of time discussing cerebellar anatomy before providing an overview of some of the challenges that are associated with imaging the cerebellum. I'll then wrap up by summarizing some of the recent advances that have been made to better understand the functional organization of this structure. So like the cerebral cortex, the cerebellum is divided into left and right hemispheres. Unlike the cerebral cortex, there's a phylogenetically old portion of the cerebellum known as the vermis, which integrates visual, vestibular, and proprioceptive information. Now, the image that I'm showing you is a post-mortem human cerebellum scanned at 200 micron resolution. And at this resolution, you can clearly make out the beautiful branching structure of the cerebellum, cerebellar white matter. And you can also see the deep cerebellar nuclei embedded within the white matter of the cerebellum. Now, one important difference between the architecture of the cerebellum in contrast to the cerebral cortex is the incredible tight folding of the cerebellum, which you can see clearly illustrated here in the sagittal slicing through the cerebellum. And this tight folding is a very important feature of the cerebellum that introduces unique neuroimaging challenges, which I'll discuss later. So the foldings of the cerebellum are called folia, and they're approximately 1 to 2 millimeters in width. And if we zoom into a folium, we can see that there are 3 distinctive cortical layers, starting with the molecular layer, which is mostly comprised of axons, the Purkinje layer, home to the major computational neuron of the cerebellum, the Purkinje cell, and then the granule layer, where the 50 or so billion granule cells reside. And this 3-layered architecture is repeated across the cerebellar cortex and is a unique feature of the cerebellum. So, like the basal ganglia, we can't talk about the cerebellum without recognizing that this structure operates within the context of a large corticothalamo-pontine circuit. In fact, in terms of a numbers game, the corticocerebellar system is one of the largest neuronal circuits in the human brain. 40 million axons leave the neocortex through the cerebral peduncles, the vast majority of which project to the pontine nuclei. From there, this stream of information is projected onto 50 billion granule cells corresponding to an expansion of 1 to 1,000. Granule cells then give rise to a single parallel fiber, and each fiber innervates 15 million Purkinje cells. Purkinje cells then deliver their output via the deep cerebellar nuclei to the thalamus and then back to the neocortex. So interpreting the cerebellum within the context of the cerebral cortex is important if we're going to further our understanding of cerebellar function. Now, while we know a lot about the anatomy of the cerebellum, the advent of functional neuroimaging really allowed us to further our understanding of cerebellar functional organization. So from the first landmark PET study that showed cerebellar activation in response to single word processing, many hundreds of neuroimaging papers have been published showing cerebellar activation in response to attention, response reassignment, higher order rule processing, verbal working memory, language, social cognition, amongst many, many others. And this research has been critically important in shaping how we think about the function of the cerebellum in humans. Now, despite this large body of work, it's important that we recognize the challenges that researchers have faced in imaging the cerebellum. From physiological artifacts to spatial normalization, there are many reasons why researchers have removed this brain region from the field of view in fMRI research. And one important factor to consider are physiological artifacts. So due to its dependence on blood flow and oxygenation, the bold signal is highly sensitive to changes in respiration and cardiac function. And this is especially true for the cerebellum and the brainstem, which are surrounded by a dense bed of vasculature, as you can see here. So as the heart contracts and relaxes during the cardiac cycle, periodic variations in arterial blood pressure are transmitted along the vasculature, resulting in relatively localized motions and deformations of the brain. And this motion clearly affects the cerebellum. But fortunately, physiological signals are easily recorded, and when they are included as nuisance regressors in a GLM, they can account for 35% of the variance around the brainstem and the cerebellum. Now, a second important factor to consider is the bold signal itself, which relative to the cerebral cortex has a very different origin story in the cerebellum. So presented here is a schematic of the circuitry of the cerebellum. The main computational unit is the Purkinje cell here in green. Mossy fibers bring input from the cortex and the spinal cord to the granule cell, which then synapses onto the Purkinje cell via the parallel fiber. And interestingly, work by Thompson and Matheson and colleagues has found that increased spiking activity of Purkinje cells is neither sufficient nor necessary to elicit cerebral blood flow responses in the cerebellar cortex. Now, what that means for the cerebellum is that it's likely that the bold signal is largely reflective of input signals from the cerebral cortex, not Purkinje cell firing. And this is an important detail to consider when we interpret task-evoked or resting state cerebellar activation. So a third factor to consider is anatomical localization, which has long been a challenge for cerebellar researchers. Presented here in this video is a reconstruction of an individual human cerebellum resolved down to the level of individual folia. And this is the same post-mortem cerebellum that I showed you a few slides ago, scanned for many hours at 200 microns. And color here indicates local curvature, with green indicating the crest of a folium. When this reconstructed volume is flattened, it is estimated at 78% of the surface area of the entire human neocortex. However, due to the current constraints of imaging techniques, it's really not feasible to resolve down to the level of individual folia in our research participants. And even when the cerebellum is resolved to the level of groups of folia rather than individual folia, a resolution higher than 200 microns is required for complete unfolding. And furthermore, the typical resolution of functional imaging data is between 2 to 4 millimeters isotropic. Therefore, the activation from adjacent folia is averaged. And you can see here in this atlas from Van Essen that there are two sites of individual finger representations in the cerebellum, one in the anterior lobe and the other down here in the posterior lobe. However, you can see here that these activations, these loci are fractionated and distributed over a number of folds of the surface. And this problem arises from the fact that the level of detail of the anatomical unfolding is simply too high for the resolution of the functional data. So what then can we do? Well, to address these shortcomings, in recent years, a probabilistic atlas has been developed, which is resolved not to the level of the folia or groups of folia, but to the level of lobules. And this development has taken the form of a spatially unbiased infratentorial template, or SUIT for short, presented here on the left in coronal, sagittal, and horizontal views. And each color denotes a different lobule. And more recently, a flat map representation of this atlas has been developed to allow for the visualization of cerebellar gray matter in one concise view. So you can see here, starting from lobules 1 to 4 in the anterior lobe, traversing them through crus 1 and crus 2, all the way down to lobule 10 in the floccular nodular lobe. And this is a really nice way to concisely visualize cerebellar activation in one view. So now you can see, if we visualize these same motor activations on the group map, the one that's unfolded to the lobules, you get these closed clusters in the anterior lobe and in the posterior lobe, compared to the fractionated clusters in the individual atlas here on the left. And furthermore, the deep cerebellar nuclei, which sit in the white matter of the cerebellum, these can also be normalized to suit space. However, the difficulty with the deep cerebellar nuclei is in identifying them clearly on a 3T scan. So they're really small structures. They're comprised of only a few hundred voxels, and they're not always visible on scans. Therefore, it's likely that scanning at 7 Tesla would improve segmentation of these structures. Now, fortunately, the challenges that face cerebellar neuroimaging can largely be addressed, and because of recent advances, we have been able to glean many important insights about cerebellar function and organization. So for the last part of this talk, I want to walk through some developments that have been made in mapping the cerebellum using resting state and task evoked data. So we know mostly from polysynaptic tracing work in non-human primates that communication between the cerebral cortex and the cerebellum is often understood as relying on a series of parallel loops, each connecting a single cortical and cerebellar region in a closed loop circuit. So for example, Strick and many others have shown that if you inject retrograde tracer into area 46 of the prefrontal cortex, that cells in the dentate nucleus are labeled. And what's important here is that these dentate neurons are located in regions that are spatially separate from those labeled after injections into motor areas of the cerebral cortex, indicating a clear motor, non-motor topography. And this labor-intensive work was done in primates, and it's very invasive. However, resting state fMRI over the past decade has provided a great alternative, a great non-invasive alternative for mapping corticocerebellar connectivity. And a lot of this important work has been done by Buckner, Yeo, and colleagues using resting state data, and they have corroborated the seminal anatomical work by demonstrating a one-to-one mapping between corresponding networks of the cortex and the cerebellum. So they generated a map of the cerebellum by regressing each cerebellar voxel against cortical resting state networks. And then they did a winner-take-all approach to assign each voxel of the cerebellum to the cortical network with which it was most correlated. And this is the resulting map that they achieved. And what they showed was that supporting evidence for connectivity between motor regions of the cortex, so in turquoise and blue here, and the cerebellum, illustrating this known somatotopy of the cerebellum. And what was really interesting, however, was that the intervening regions between these motor representations, so all these regions here in pink and in purple, these were largely mapped to cortical networks associated with non-motor function. So these pink and red networks in the association regions of the cortex. And more recently, work from the same group has also shown that this corticocerebellar connectivity can in fact be identified at the individual level, shown here in 2 example subjects, and that the individual cerebellar topography contains multiple representations of cortical network, similar to what was shown at the group level. So, if functional parcellations of the cerebellum are to eventually be used for translational or clinical purposes, it really is important that this functional organization can be identified in individual cerebella, as shown here. Now, task-evoked fMRI activity has also been leveraged to derive a functional parcellation of the human cerebellum. And what's notable about task-evoked fMRI is that it allows for semantic meaning to be assigned to functional subdivisions by regressing cognitive features against task profiles. And recent work has found that, for example, regions in the anterior lobe of the cerebellum encode left and right hand motor presses, a known somatotopy in the cerebellum. Another region encodes saccades and other features related to visual recognition. A fourth is a motor region that's driven by action observation. But interestingly, beyond these motor regions, the features most descriptive of cerebellar functional organization captured a range of cognitive processes from attention to narrative, verbal fluency, and autobiographical recall. And this nicely illustrates this sort of rich functional organization of the cerebellum. And similar to the resting state work, these task-evoked parcellations can also be reproduced at the individual level. And while it's largely a practical decision to go to the group, a lot of information is lost by averaging over individual subjects. So one interesting methodological question for future fMRI research and MRI research will be to determine how much data, resting state or task evoked, is sufficient for deriving functional parcellations at the individual level. And another way that cerebellar researchers think about functional organization is in terms of functional gradients. So in this recent work, diffusion map embedding was applied to resting state data from the Human Connectome project to show that cerebellar functional regions follow a gradual organization which progresses from language and default mode network regions here in red to these motor regions in blue. And then a second gradient was identified that isolated working memory, and frontoparietal network areas. And all of this work, be it functional parcellations or functional gradients, contribute to our understanding of cerebellar functional organization, both by itself and within the context of the cerebral cortex. And finally, making these maps and these gradients available is an important step in advancing the field of cerebellar neuroimaging, because they provide a useful guide in designing studies to test specific functional hypotheses, as well as facilitating new ways to interpret cerebellar activation. So here you can see a brain viewer has been created for visualization of task maps and parcellations, and you can scroll through each of these different maps. You can also download them for your own research. And then the functional gradients have also been made available for viewing in an interactive fashion, and they can be accessed here. So overall, despite the challenges that face cerebellar neuroimagers, I really do believe that it's a very worthwhile endeavor to use MRI and fMRI to study the cerebellum. There's a lot to be learned about this mysterious and complex little brain. So with that, I would like to thank my PhD advisor, Richard Ivory at UC Berkeley, my master's advisor, Jorn Diedrichsen at Western University, and all the members of the Cognition and Action Laboratory at Berkeley who've been a great support in my graduate career. Thank you.